students. Let us learn about cloning vectors, selectable markers, and insertional inactivation today. I hope from the previous videos you have understood what are the steps that we have been doing in order to get a recombinant DNA. So what all were the steps that we covered till now? Let us just quickly recap so that we can go ahead with understanding today's concepts. First, we identified a gene of interest in one of the organisms, and we had to take out that gene of interest, which we cannot do in an isolated way. We had to extract the entire genome of that organism, which we did using the step DNA extraction. Once we got our DNA, that is not enough. We had to cut it. How did we cut it? We used restriction enzymes for that. Once restriction enzymes cut the DNA or the genome, we had to elute it out on a gel electrophoresis unit. I hope you have understood all these steps from the previous videos. If you haven't watched the previous videos, please go through that so that you get an idea of what we are talking about. Okay, now we have our gene of interest, which is only one copy, which is not enough for us. We need to have multiple copies of that gene of interest. How do we get that? We used a technique called as polymerized chain reaction or PCR, and we got multiple copies of our gene of interest. All right, let's start understanding today's concept now. Since we have our gene of interest, let's understand how a recombinant DNA is made. All right, this can be done in two different ways. One is for me to take the gene of interest and put it in the host organism like so, using direct methods. Let us say this is the nucleus of that organism. This is a bacteria, so it doesn't have a nucleus. However, if let us say it is a plant cell or an animal cell, I am able to directly insert my gene of interest over here. Let us say, children, here we have our gene of interest inserted into the genome of another organism like a plant or an animal using direct methods. When I say direct method, what do I mean? I mean that there is no delivery vehicle called as a vector being used here. I directly inject my gene of interest into the genome of a plant cell or an animal cell. So our recombinant DNA would look something like this. This is the genome of the organism into which somewhere I have inserted my gene of interest over here. That is how it is going to look. And for that, I have different methods that I can use. These students, what are these direct methods of creating a recombinant DNA? In that comes microinjection, gene gun, and disarmed pathogen. What happens in these three cases? We are not using a delivery vehicle like a vector. Instead, we are allowing the gene of interest to directly get into the nucleus. So let us say this is the nucleus of the host. It will have its genome definitely. Into this, we are injecting our gene of interest. So what are the ways in which it can be done? First is micro injection, wherein you use micro pipettes like so. A pipette would look like this. Using an electron microscope, you are observing the cell and then injecting our gene of interest into it. That is called as microinjection, usually done for animal cells. Usually done for animal cells. The next one is gene gun, also called as biolistic method. In this, what are we doing? We are coating microparticles of gold or tungsten with our gene of interest, like so. Then we are making this microparticles to move at high velocity and bombard your genome of the host. These ones, the microparticles which are already coated with the DNA of interest, gene of interest, at very high speed, they are going and bombarding the genetic material of the plant cell. In that case, this DNA would get integrated. The third one is disarmed pathogen. So what is a disarmed pathogen? Disarmed is the ability to cause a disease of the pathogen. That part is removed from the pathogen. That is what is meant by the by disarmed pathogen. So let us say you have a bacterial cell which usually gets infected by bacteriophages. You know what are bacteriophages? They are viruses. They have the ability to infect your bacterial cell. What if this pathogenicity, in other words, the ability to cause a disease is removed? Then this can inject our gene of interest into the bacterial genome. So those are the three direct methods. Now, direct methods are expensive to a large extent. Usually, we use vector based transfer of genetic material into the host, which I will be explaining in a minute. So, what is a vector? Vectors are delivery vehicles. Alright, this can be either a plasmid or a bacteriophage. It can be either a plasmid or a bacteriophage. So you should know what is meant by a plasmid, isn't it? So I have drawn here one bacteria. This is, let us say, one bacteria. Bacteria will have its genetic material. We'll call that as a chromosomal material. Yeah, its genetic material we are going to call as chromosomal material. Other than the chromosomal material, some DNA will be there in bacteria. Circular DNA will be there in bacteria, like so. Those are called as plasmids. So how do we define a plasmid? Plasmids are extra chromosomal, meaning other than the chromosomal material, whatever is present, extra chromosomal, circular DNA, which is naturally occurring in bacteria or prokaryotes. Those are called as plasmids. Why is it of any significance to us? It is of significance because this can also be easily cut by using restriction enzymes. I hope you remember when we learned about restriction enzymes, the diagram that we looked at had two things in it. We'll just go back so that I can recap it for you. The action of restriction enzyme, when we looked at what all did we see? We saw we were cutting two things at a time. Even though we didn't discuss it at that time, I just told you that it has the ability to cut at special sequences called as recognition sequences. All that I said, but the diagram had one more thing in it. That is this. This was the gene of interest for us, and this part is called as a vector DNA. At that time, I didn't discuss it because we had not covered it, right? So here, whatever restriction enzyme you're using to cut our gene of interest, the same restriction enzyme we are going to cut our vector also, our plasmid also, we will cut using the same restriction enzyme. For what purpose? So that we get the same complementary sticky end on the vector also, as well as the gene of interest also. So this both can fit into each other. This is the sticky end of the vector, let us say, and this is the sticky end of the gene of interest, let us say. They both tie into each other very neatly, and then this can be sealed using another enzyme called as ligase. This process is called as creating a recombinant DNA. Hope that is clear. All right. So for this process, we need to have a vector which can deliver our gene of interest into an organism. We're not done. Let us say we have started doing that work. All right. Now, let us say I have with me some plasmids, plasmid meaning extra chromosomal circular DNA, which I have cut with the restriction enzyme. The same restriction enzyme that I used for cutting what? Our gene of interest also. So this is your gene of interest, which I have cut with restriction enzyme. This is the plasmid. So I integrate the gene of interest onto the plasmid, right? This whole thing I transfer into my host. So what is this host now? Host has got a recombinant DNA. So we'll call this host as recombinant host. recombinant host or recombinant. Clear? Students, one thing that you need to keep in mind while understanding the entire concept of recombinant DNA technology is that we don't start our recombinant DNA procedure using just one copy of our gene. We make multiple copies of our gene of interest. We make multiple copies of our plasmid. We should have multiple number of cells, many number of the host cells. Clear? Why? Because this integration of our gene into the plasmid is a chance factor. The plasmid, entire plasmid and the gene coming into the host is again a chance factor because you are using certain tools for this. Okay, so all the end product cells, host cells, may not have plasmids and gene with it. Some will be plain host cell only. Nothing else would have got integrated into it. On the other hand, some host cell would be having only the plasmid with it. Let's say only this plasmid has integrated into it. All right. Some will have host cell will have plasmid also, gene of interest also. So how to select it out? How do we know which all cells have got our gene also, plasmid also? Only if the plasmid is there, gene is there. But in some cases, only the plasmid would get integrated. Plasmid would not have integrated the gene of interest into it in the initial step. So in order to select out, we need to have a marker. A marker is something like a indicator. Indicator in chemistry, you must have used some indicators which gives a particular color and so on. Similarly, here also we should have an indicator which tells you, yes, this is a host cell which has got the plasmid plus the gene of interest. Usually, these markers are genes that are present on the plasmid itself. Clear? Yes. So we are going to understand that part of that part called as selectable marker. Like I already told you, selectable markers are usually indicators or markers which are present on the plasmid itself. So usually one of the properties of a good vector is the presence of a selectable marker. About this vector called as PBR 322, we'll be covering up in the next video. Right now, just understand that we are looking at a vector called as PBR 322 in which two genes called as ampicillin resistance and tetracycline resistance are present, and these are the selectable markers. So we are also learning an answer to the question, how does antibiotic resistant gene act as a selectable marker? So please be attentive, you should be able to understand this. All right. So usually you know what is an antibiotic? Something that kills growth of bacteria. So normally on a medium in which you provide nutrients, definitely our bacteria will grow. However, if I have a nutrient medium into which I have added some antibiotics, let's say the name of the antibiotic is ampicillin, and the other nutrient medium into which I have added antibiotic tetracycline, in both cases, the bacteria is not going to grow. However, let's say I have a host organism which is a bacteria here into which our PBR 322 is integrated. Yeah. So this plasmid has a gene which is going to produce a protein which is going to give resistance to this antibiotic also, this antibiotic also. So on both these plates, both cases, our bacteria is going to easily grow. However, so that can act as the first set of marking, let's say. But we are not stopping there. Okay, we are not doing that. Instead, we are doing a little more trick onto this. So what are we doing? We are inserting our gene of interest into the plasmid initial step, into the plasmid in one of these genes itself. So what will happen to this gene? This gene is a tetracycline, which is an antibiotic resistant gene. Tetracycline resistant gene, which will give resistant to tetracycline antibiotic on the medium. Right? However, to that gene sequence, let's say the gene sequence is GATATG, is the gene sequence of tetracycline resistance product. The gene is here. This is going to give the resistant protein, let's say. Into this sequence, I am inserting my gene of interest here. Alright. So what am I adding? C, G in between. What does it do? It will inactivate this. Isn't it? It will inactivate the tetracycline resistant gene. This is no more the gene sequence that we need for producing the protein against tetracycline. We have some other sequence G, A, T, C, G, A, T, G. It is useless. Right? Our gene of interest is here, which we need for the recombination. But in this case, that against tetracycline, nobody is going to give this bacteria any resistance. Alright, why are we trying to kill it? In order to select it out. So how is the selection done? Please be attentive. On a nutrient medium, when you try to grow, it will grow easily. On this nutrient medium, our host will grow. Okay, this step we're not doing just for you to understand, I'm telling. On the ampicillin resistant plate, that is nutrient medium having ampicillin antibiotic, our plasmid is there in it, na? The host will easily grow because this plasmid is giving resistance to it. Now, on this medium, it is not going to grow. Why is it not going to grow? Because the tetracycline gene is already inactivated by insertion of our gene of interest. Clear? So, what do? I try to take few colonies from this plate. Right? On this plate, whatever is growing, if it is growing on this plate, having antibiotic ampicillin, it shows that my host organism, which is a bacteria, is able to resist this ampicillin because of the presence of my plasmid. Alright. So from all these colonies, I'm going to take one speck. From here, I'm going to take one speck. I'm going to plot it here. Allow it to grow. I'm going to take from little bit speck from here and trying to put it here and trying to grow. Whatever grows on it shows that our gene of interest is not there. Whatever grows on it is useless for us, meaning it's not a recombinant one. It does not have our gene of interest. However, let's say I take one from this colony. From this colony, I take a speck and I try to grow here. Nothing is growing, let's say. Right? Nothing is growing here. It shows that this colony is having cells which has got the plasmid also, the gene of interest also. I hope that is understood.